Hey, so I thought I would do a quick video on the anatomy of the eye model since it does look a bit different than the dissection. So this is a superior view of the eye. One of the ways you can tell that is just by looking at the eye itself. And so it has 6 extrinsic eye muscles. 4 of them are rectus muscles, 2 of them are oblique muscles, and the superior oblique attaches to the eye via a tendon, this white structure, and then it hooks around this little pulley called a trochlea and then goes back. So the muscle itself looks like another rectus muscle, but because its direction is changed by this pulley, it's still an oblique. It moves the eye and it rotates the top of the eye medially towards the nose. This also helps me know which way I'm looking at. So like this trochlea is always on the part of the orbit that is medial or closer to the nose. Now, the 4 rectus muscles, this is the superior rectus, the lateral rectus, the medial rectus, the lateral rectus, and the inferior rectus. The superior oblique is harder to see. I can kind of rotate this up and around, and we can see the inferior oblique here coming right in. And where it attaches to the eye, it's very fleshy, and all of the rectus muscles attach with a very fleshy attachment. It's only the superior oblique that attaches with this tendinous kind of connection here. So if I take these little muscles off, we can see a bit more. Now, looking at the eye itself, actually real quick before we go forward, you can, if I rotate it up, see this, and this is the lacrimal sac, and that hole right there is one of the lacrimal puncta that allow the lacrimal canaliculi to drain tears into the sac, which then goes into the nasolacrimal duct and drains into the nasal septum, or nasal cavity, excuse me. So when we look at the eye itself, the outer layer of the eye is the fibrous tunic, and that includes the sclera, the whites of our eyes, plus the cornea, the clear portion up here, plus the lens. Now coming out of the posterior aspect of the eye is the optic nerve. So then this runs back through the optic canal to the optic chiasm. Now on this particular model, we have a section of the fibrous tunic, specifically the sclera, that's been cut out, showing us the medial vascular choroid, which is the next layer down, and that's part of the vascular tunic, or vascular layer of the eye. And all the way in, we'd find the retina, which is part of the neural tunic of the eye. Now, if I take this eye, and I can take the top half off of it, so when I do this, the cornea is left behind on this model, and so we're able to see the iris, which is part of the vascular tunic, and then this is the sclera, part of the fibrous tunic. Now, in an eye itself, and they kind of represent this with a little ridge, this layer of the fibrous tunic has an extra epithelium on top of it called the bulbar or ocular conjunctiva, and then the inside of the eyelid would have the palpebral conjunctiva on it. It's not really differentiated here other than by this line, so nothing really crazy on the model. All right. So here we're looking at the inside of the eye, and the eye, remember, it has 2 big spaces on the inside. So it has the posterior cavity, which is this big region right here that I'm going to lift out right now. Hopefully the lens stays in. And the anterior cavity, which includes the space, so this is the cornea that I'm lifting out, part of the fibrous tunic. And the lens right here, also part of the fibrous tunic. So the anterior cavity of the eye is this space, and the anterior cavity has 2 smaller cavities in it. The anterior chamber, which is in front or anterior to the iris, and the posterior chamber, which is behind or posterior to the iris. So I know it's confusing, but remember, chamber is like a little room, and so cavity is a big space. So the big posterior cavity, the big front cavity, anterior cavity, and in that anterior cavity we have the small front room and the small back room. So the anterior chamber and the posterior chamber of the anterior cavity. You can also see the tunics well in this view, and so we have the fibrous tunic, which is superficial. The red is the vascular tunic, and we don't really see the retina as a distinguished color on this, but usually it's represented as like yellow, kind of like the yellow of the optic nerve that we see here. So there is a neural tunic here, it's just not painted in on this model. We can see on this model where the optic nerve exits, and that little space there is not going to have any photoreceptors. So we call that the optic disc, and that's what causes your blind spot. The retina would extend up here, and they did draw the blood vessels, even though the retina is not really shown. And the retina is kind of like a clear-ish tissue. It doesn't really show up that well on our dissections. If you look at the cow eye dissection video, you can see it's kind of like a slightly opaque yellowish color, almost like the color of the stapler. And it's a very thin layer that sits on top of the vascular tunic, or the choroid, when we're back behind the retina. So it is kind of indicated here by the vessels that you can see. They just didn't make it like highlighted by a different color. Now, the retina extends anteriorly in the eye up until this little black serrated line called the ora serrata, and then that's where the retina stops. So on the retina, we would find a concentration of cones, the ones that allow you to see color right here in the macula lutea. And then there's this divot, which I'm not sure if it shows up well in the video, but you can kind of see a little divot here. That's called the fovea, like pit, fovea centralis. And that's where you have the highest concentration of cones. So when you look at something, it projects the image right onto that spot. Wherever you look, what you're focusing on is sent here, and then your peripheral vision comes from the other parts of the retina. The further away from the fovea centralis that you get, the more rods you have and the less cones. So you get better like night vision, and they're more sensitive, but they aren't able to distinguish colors as well. Well, not at all. They don't distinguish color. They just kind of show the intensity of the light source. Whereas your cones allow you to distinguish between 3 wavelengths of color, allowing you to then differentiate color in your vision. As the axons from the ganglion cells that came from the bipolar cells that came from the photoreceptors exit, they're then going to travel on the surface of the retina until they reach the optic disc and then dive back through the optic nerve. Again, why we don't have photoreceptors in that region. Now, as I follow the vascular tunic, which is the bulk of the red here, remember the retina is just a thin layer on top. So the vascular tunic, and this is specifically the choroid. Now, once I get to the ora serrata, we're going to have this get thicker, and there'll be muscle in here. That's called the ciliary body, this whole region. You have the ciliary muscle in here, and then you'll have the ciliary processes. From those ciliary processes, the suspensory ligaments go out and attach to the lens that would be sitting right in here. And that way, when the ciliary body is either pulled away or relaxed inward, it will stretch the lens out, making it thinner and refract light less, or relax the lens and it gets fat again, and then that bends light very well for looking at things up close. So again, the suspensory ligaments are what anchor the lens to the ciliary body, specifically this little ridge of processes. And on those processes, they will be secreting aqueous humor that then flows out around the lens and then through the pupil into the anterior chamber. And eventually you have little pores down here that lead to the scleral venous sinus that will reabsorb that aqueous humor. The posterior cavity here was full of vitreous humor, and that exchanges, but very slowly. So like it takes weeks or months to exchange the fluid that's contained within that. The protein in there that makes it like vitreous, that makes it kind of like a loose jello, that though is built when you're in utero, and it's kept your entire life. Part of why the eyes don't really change size. So like kids' eyes look so big because they're basically adult eyes slapped into a kid's face. I think that covers it for the anatomy that we have on these models. I hope that helps.